Hello, everyone, and welcome back to the Mid-Atlantic Micro Nano Alliances virtual seminar series. I'm MAMNA co-president Ryan Sokol, and we're joined today for the first time by our MAMNA treasurer, Oliver Barham. Oliver, who is our presenter today?
Hi, everyone. Today, we're joined by Ambika Somasundar, who's presenting on her research.
Hello, everyone, and welcome.
In the area of active matter and colloidal motors. Ambika is a fourth-year chemical engineering grad student at Penn State. She works under the advisement of Dr. Ayusman Sen and Dr. Darrell Velegol. Previously, she received her BS in chemical and biomolecular engineering from the National University of Singapore in 2014. In addition to her role as a grad student, scientist, and researcher, she also practices and performs Indian classical dance. Ambika, take it away.
Thank you, Oliver, for that introduction. Hi, everyone. So today I'm going to talk to you a little bit about positive and negative chemotaxis of enzyme-coated liposomes. The objective of this work is to try and understand the question of how can we steer soft colloids and protocells in both positive and negative chemotactic directions. In particular, today, I'm going to discuss and rule out existing mechanisms of transport for these colloidal motors and allude to solute-phospholipid-based interactions as a potentially new and previously unrecognized mechanism of transport. But why do we care so much about directional migration? The ability to move towards or away from specific chemical signals is crucial for the survival of biological systems. Directional migration enables cells to move towards fuel, away from toxins, and coordinate collective behavior such as tissue formation and biofilm formation. Here is an example of a vesicle that is being transported across a cell through the chemical conversion of ATP and the conversion of chemical energy into mechanical motion. Here's another example of a white blood cell that is seeing a foreign particle in its environment, sensing it and moving towards it, and ultimately engulfing it. So these kind of movement is called chemotaxis, and it is in two general directions. So it is called positive chemotaxis when the cell is moving up the gradient, up the chemical gradient, or towards higher concentration. And it's called negative chemotaxis when it moves down the gradient or away from the high concentration of chemical. Inspired by nature and biology, scientists all over the world have tried to create a variety of synthetic material that is able to show this directional migration. So among the 4 studies that I show you here, whether it is enzymes or enzyme-coated particles, or catalyst-encapsulated soft particles, all of them show positive chemotaxis. That is, they move towards higher concentration of chemical. In the motors community, negative migration of enzymatic systems has not really been observed. So more recently, the motors community has moved from inorganic motors such as gold platinum nanorods to soft colloids such as liposomes and polymerosomes. And this is because of the variety of advantages that they pose. So some of the advantages is that they are highly biocompatible, so they can serve as cargo delivery vehicles. And also because of their high resemblance to the membranes of natural cells, they can be artificial cell models to study a variety of things. It could be origin of life studies or to how a virus infects a cell. In this work, what we wanted to do is see how liposomes that are made up of phospholipid molecules, when they have enzymes attached on them, how would they move in gradients of substrate? So similar to previous studies, we hypothesized that enzyme-coated liposomes would show positive chemotaxis. That is, they would move towards higher concentration of chemical. In order to test this hypothesis, we employ mainly 3 techniques. So the first technique is thin film hydration for the liposome synthesis and extrusion to form uniformly sized liposomes. So here are the 3 lipids that I show you here is the lipids that we use to make our liposomes. So the first lipid has a rhodamine molecule. So the rhodamine has fluorescence characteristic, and we're able to see it under the confocal microscope. The next lipid has a biotin head. So this gives us the functionality to attach enzymes onto the external surface. And the third phospholipid that I show here is the backbone phospholipid that makes over 95% of the liposomes. So once we form these uniformly sized liposomes, we want to attach enzymes on them. And we do this through this biotin-streptavidin conjugation method. We first biotinlate the enzyme and then attach streptavidin on it, and then mix it with the uniformly sized liposomes to get enzyme-coated liposomes or active liposomes. So after we form these active liposomes, we want to test their chemotaxis in imposed gradients. And we do this through a 3-inlet, 1-outlet microfluidic channel. Typically, we flow the fluorescently tagged liposome through the mid-channel, and the side channel has buffer. This is the control. So we take one of the side channels and replace the buffer with substrate, and that constitutes our experiment. We compare the experiment with the control to see how it moves in these imposed gradients. So this is a typical graph that we get out of this 3-inlet, 1-outlet microfluidic channel experiment. So there is a laser scan that occurs from the confocal microscope at the end of the channel. And this allows us to scan how the liposomes move from the mid-channel to the side channel. So like I said, in the control experiment, it would be through Fickian diffusion. And in the experiment where we replace buffer with substrate would be where we observe how the liposomes move towards the substrate. So the first enzyme that we tested was catalase. So catalase is an enzyme that converts hydrogen peroxide into water and oxygen, and it's pretty fast. And similar to literature, we were able to see that active catalase-coated liposomes also move towards higher concentration of hydrogen peroxide. And this was in agreement with enzyme molecules and enzyme-coated hard particles, as we saw from literature. So no surprise there. The next enzyme that we tested was urease. Now, urease molecules, as well as urease-coated hard particles, have been shown to also show positive chemotaxis in gradients of substrate. So it was very surprising for us when urease-coated liposomes actually moved away from high concentrations of urea. So in order to confirm that the lipid vesicle itself does not have any effect in presence of urea, we tested inactive liposomes, that is liposomes without enzymes in gradients of urea, and we see no negative movement. So this led us to conclude that the product of the urease reaction is probably interacting somehow with the lipid vesicle and is causing the negative chemotaxis. So this is what we tried next. We subjected lipid vesicles, both active, here I show only inactive liposomes, but we subjected both inactive and active lipid vesicles to a gradient of ammonium carbonate. And indeed, we see a negative chemotactic migration. Not only does ammonium carbonate show this negative chemotactic behavior, but a variety of salts and solutes is able to make the liposomes move in the negative chemotactic direction. So that gave us a clue that there is an additional mechanism of motion that is acting on these soft lipid particles. So that brings me to the next question of this talk. What exactly is the mechanism of this negative chemotaxis? And how can we control it to make these lifelike adaptive materials? So for the rest of the talk, I'm going to be discussing how lipid vesicles that without enzymes act in various gradients. We had a couple of hypotheses that we wanted to test, and spoiler alert, we were able to rule them out, and our current standing hypothesis is a Hofmeister series-driven solute-phospholipid interaction-based chemotaxis. So the first mechanism is diffusiophoresis. Diffusiophoresis is a mechanism by which particles are able to migrate in gradients of electrolytes or non-electrolyte. For today, I'm going to discuss electrolyte diffusiophoresis, that is salts, migration in salt gradients. So salts have cations and anions, and both of them have differences in diffusivity. And due to the differences in diffusivity, there can be the generation of a spontaneous electric field. And if the particle has charge, a zeta potential, it would move in this spontaneously generated electric field. So in order to rule out diffusiophoresis, we took two salts, ammonium bicarbonate and sodium chloride, which would form opposing electric fields. And we tested the movement of the lipid vesicles in these opposing electric fields. And we found out that ammonium bicarbonate moves one way, but in sodium chloride, it doesn't move the other way. And so we can effectively rule out the diffusiophoretic mechanism. The next mechanism that we wanted to test was density-driven flows. So what we did for this is we replaced the liposomes in our experiment with solid particles. So solid 200 nanometer polystyrene beads have higher density. And if there are any density effects, it would feel it more, and we would be able to see a shift at the interface of the solute and the particle. However, we don't see this shift, and we were effectively able to rule out density-driven flows. The next mechanism that we had in mind was osmophoresis. So osmophoresis is a mechanism of motion when a membrane-bound colloid is placed in a gradient of impermeable solute. Here, what happens is when, due to the difference in osmotic pressure inside and outside the vesicle, water is expelled from the side facing the high concentration and is admitted in the opposite side, propelling the lipid vesicle forward. Here, what we did is we compared our experimental values of negative chemotaxis with theory and found out that we were able to get an overlap only with a huge factor of 10 to the power 4. So this told us that osmophoresis is negligible in our system, and we can effectively rule out osmophoresis. So at this point, we tested all the phoretic mechanisms that were available, but we weren't sure what was happening. And so we tried to dig in literature more, and that's when we came across the Hofmeister series. So the Hofmeister series is a series of anions and cations according to their ability to salt out or salt in organic molecules. Now, salting out means that the ions are able to aggregate the protein in solution, and salting in means that the ions are able to solubilize the proteins in solution. So the Hofmeister series has been around since the 1800s, but nobody understands how it completely works. And there are a few factors that affect how these ions work. And these can be due to size and shape of the ions, hydration thermodynamics, charge distribution, hydrogen bonding, and hydrophobicity. So when you go from left to right in this Hofmeister series, you can see that the salts on the left are well hydrated, and on the right are less well hydrated, that is poorly hydrated. So well hydrated salts do not associate with the hydrophobic part of biomolecules and repel them, while the poorly hydrated salts are able to associate with the hydrophobic part of biomolecules and is attracted towards them. What was interesting is that the liposomes that showed negative chemotaxis, they showed negative chemotaxis in salts that were in the left side or the well-hydrated region of the Hofmeister series. So in order to test our hypothesis of whether the liposomes show movement according to the Hofmeister series, we took a salt from the other end of the Hofmeister series and tested it. And indeed, we get a reverse positive chemotactic effect. So well-hydrated salts, such as the bicarbonate ion, they are depleted from the lipid-water interface, and the liposomes are repelled by them. Whereas for the poorly hydrated salts like thiocyanate, they are enriched at the lipid-water interface and thus are attracted by the liposomes. So basically, the ion depletion or ion enrichment at the lipid-water interface is it shows positive or negative chemotaxis. And so in summary, we were able to show that active and inactive liposomes move both in positive and negative chemotactic direction. We were able to effectively rule out diffusiophoretic, osmophoretic, and density-driven transport mechanisms. We showed that these liposomes move according to the ions in the Hofmeister series. And this is a first step in programming directional motion in adaptive lifelike material. With that, I'd like to thank my advisors, Dr. Ayusman Sen and Dr. Darrell Velegol, my colleagues and collaborators, Drs. Shubhadeep Ghosh, Farzad Mohajarani, Paul Kremer, Ting Lu, and Linisha, and also the members of my lab, and the funding source that made this research possible, and as well as MAMNA for giving me this opportunity to share my research. Thank you.
Yeah. So that was great. It's so weird, I think. I'm still not used to it, like giving a talk and having the talk end and like, it's, it's like silent. I don't know. I feel like it's like a little bit less fun than, like a typical talk. But that's really, really, really great work. I mean, you obviously know that in terms of getting published in Nature Nanotech. So we have some questions from the chat. And so our first question comes from Heidi Leonard. And the question is, for positive chemotaxis, is it possible to control the degree of enzymatic conjugation for the liposomes?
That's a good question, actually. So for the lipid vesicles itself, we have only 2% of biotin. And 2% of biotin, and only that much is covered by enzymes. So if you want to increase the enzyme coverage, you just have to simply increase the biotin concentration. So from 2%, you could go to 5% or 10%. But also here, one point that I would stress is that the lipid vesicle itself is 100 nanometers. So there's only a certain amount of enzymes that can actually be attached onto the surface. So for one of the enzymes that we tested, it was urease. Full coverage is only 50 enzymes per particle.
Great, thank you. So I guess probably one of the questions that people might have would be, what are the potential applications of these liposomal motors? How would they be used in practice or that kind of thing?
So lipid vesicles are very biocompatible. So they have a potential to be drug delivery vehicles in the body. And one example that I would give is like cancer cells, they have high concentration of hydrogen peroxide. So if we're able to make these lipid vesicles contain some cancer drug and catalase on their external surface, we would be able to deliver them to the cancer site.
Great. So we have a couple more questions from the chat. So this first one is from Walter Smith. And the question is, can you explain again why the ions at opposite ends of the Hofmeister series give opposite chemotaxis?
Sure, yeah, I can do that. All right. So when you look at the Hofmeister series, on the left side, so bicarbonate, carbonate, sulfate, they're very well hydrated. So they have a very good water structure around them, and they don't want to interact with the lipid vesicles. And so they kind of repel from the surface of the lipid vesicle. But thiocyanate is poorly hydrated, and it is able to attach to some parts of the lipid vesicle, and hence it is attracted, or the lipid vesicles are attracted to the thiocyanate. Does that?
Great. So our next question is from Shinmei Isanki. And the question is, how do you plan on expanding this, if you are, towards drug delivery for cancer applications? And great talk is in there too.
So definitely if we have to put it in the body, we would need many more, I mean, all the experiments that we did here were in the lab, in vitro. So definitely if we can have some collaboration with groups that do cell work, and maybe if we can put it inside cells, that would be great. But at the moment in our lab, we don't do that. All the studies are in vitro.
Great. Another question I had was, does anything other than the salt affect the movement of the liposomes?
Yes, definitely. So actually, when we were trying to disprove, rule out, I would say, osmophoresis, the solute that we used here was glucose. So even other well-hydrated molecules are able to show this negative chemotaxis. But going towards the other side, we actually saw that some amino acids are able to show positive chemotaxis of the lipid vesicles. And we're still exploring that and trying to figure out what exactly is going on.
Great. And then last question, I think. Oh, sorry, not a last question. So this one's coming straight from the chat. So this work has been done with different ions in buffer / water, I suppose. What differences would you see if you had an organic solvent?
Organic solvent like?
I'm not sure. That's what's
like,
in the...
Okay, so many organic solvents actually would just dissolve the vesicles. So since the vesicles are amphiphilic, they have hydrophobic and hydrophilic head and hydrophobic tail. And so even in like a little bit of ethanol in water, you would completely dissolve the lipid vesicles. And there's no question about enzymes. And enzymes are so easy to denature. Even if you have a little bit of ethanol in solution, you're going to denature the enzyme and you probably won't see this behavior.
Great. All right, so then I think then this will be the next one will be the next actual last question, unless we get someone else from the chat. So the question is, do other soft material colloids have similar behavior to what you've shown here?
The interesting thing is we have not seen this behavior for enzyme molecules itself. And that is something which is very interesting, because what is so special about these phospholipids that no other soft material is not able to exhibit the same behavior.
Interesting. All right, great. Well, thank you so much for your time.
Thank you so much. Yeah.
It was really great and super informative. Thank you everyone for tuning in. Oliver, do you want to announce our speaker for now?
Yes, I also want to say thank you to Ambika for your very interesting talk. And next week, for those of you tuning in, we're going to have something very different. We're going to be talking about very small, very long-lasting power sources. One of my colleagues from the Navy, from NSWC Indian Head, is going to be talking about beta voltaic power sources, which are potentially very small sources that use beta decay, a form of radiation, to generate power for extremely long lifetimes, tens of years, for decades. And they can be very small and they can be integrated into microsystems. So that's what we'll be talking about next week here on MAMNA. Bye, everyone.